Welcome to the Aspirational Neuroscience Journal Club. This time we are covering this 2021 Nature paper titled Structure and Function of a Neocortical Synapse by Holler et al. This is a truly fantastic paper. In fact, I believe it is one of the most important papers we will cover, since it directly addresses our central question: can neuronal function be predicted from preserved structure? The paper specifically asks whether the physiological strength of an excitatory cortical synapse can be predicted based on the size of that synapse as imaged by electron microscopy. This physiological strength can be defined as the postsynaptic current arising from a presynaptic action potential. And their key finding, we find a linear relationship between the synapse size and strength, providing the missing link in assigning physiological weights to synapses reconstructed from electron microscopy. In the conclusions, the authors spell out the implications of this result regarding the feasibility of interpreting and simulating large-scale connectomes. Let's jump right into the details. For each experiment, the authors decapitated and quickly dissected out the brain of a mouse, and then vibratomed a 300 micron thick coronal slice through the somatosensory cortex. Now, viewing the slice under a high-powered differential interference contrast microscope, they patch clamped the cell bodies of two pyramidal neurons in close proximity to each other in layer 2-3. Given the close proximity of the two neurons, there was a reasonable chance they would be connected by one or more synapses. The authors tested this by injecting current into one neuron while listening to the other neuron. For example, a current injected into the right neuron here created an action potential in that neuron. After a very short delay, the left neuron would respond with a small positive voltage blip on the order of 2 millivolts. This blip is a telltale sign that one or more synapses exist between the two neurons. So we can label the neuron on the right as presynaptic and the neuron on the left as postsynaptic. Here's a crude drawing to make this clearer. The researchers caused the presynaptic neuron to fire over and over again at a slow rate and watched the response of the postsynaptic neuron each time. The fact that the firing of the presynaptic neuron typically caused a positive voltage blip in the postsynaptic neuron implies that they are connected by one or more synapses. But it's important to understand that the researchers cannot see whether this is one or many synapses. They cannot see what sizes the synapses are. In this standard electrophysiology measurement, you are essentially flying blind with respect to the physical structure of the connection. Here is a plot of 10 such excitatory postsynaptic potentials, EPSPs, plotted on top of each other in gray along with their average in black. This type of electrophysiological experiment, where the functional strength of a synaptic connection between two neurons is measured, has been done for years. In a sense, this is the ground truth of the functional strength of connection between two neurons. Hundreds of such EPSP responses are collected and a histogram is plotted, along with a measurement of the recording noise, as shown here. You can see that the EPSP response is not always the same, even when the recording noise is taken into account. There's a lot of detail in this type of histogram. It shows that there is an underlying randomness to the actual process of synaptic transmission. Over the years, two competing theories have been put forward to explain these results: the one synapse, one quantum hypothesis, and the multivesicular release hypothesis. In the one synapse, one quantum hypothesis, each presynaptic bouton can only ever release a single synaptic vesicle of neurotransmitter, and when it does, the glutamate released activates all AMPA receptors present on the postsynaptic membrane. In contrast, the multivesicular release hypothesis, each presynaptic bouton can have multiple independent release sites, and the glutamate from any vesicle released can only activate the AMPA receptors directly opposed to that release site. Some previous experiments in the literature seem to support the single release site hypothesis, but others supported the multiple release site hypothesis. The authors of the Holler et al. 2021 paper developed a method to precisely image all the synapses between the recorded neurons via electron microscopy. By doing so, they showed that previous papers supporting the single release site hypothesis which relied only on a light microscopy, were in error because they were overcounting the number of synaptic connections. The Holler et al. Paper provides strong evidence for the multiple release site hypothesis, and in this process, they show how the physical size of a synapse is related to its physiological strength. The authors were able to achieve this visualization of synapses between the recorded neurons by including the molecule biocytin in their pipettes. When the electrophysiological recording session is complete, the 300 micron thick slice is placed in a fixative solution that cross-links proteins, locking the biocytin into the target cells. The authors put this fixed slice into a series of more and more concentrated sucrose solutions, and then they freeze the section using liquid nitrogen and immediately rewarm it. This is done to permeabilize the cells so that later chemical stains can penetrate easier. This freeze-thaw method may seem harsh, but it's actually considered better for preserving ultrastructure than detergent-based permeabilization methods. This whole process is described beautifully in this 2018 Journal of Visual Experiments article. The authors then immersed the section in a solution containing streptavidin HRP conjugate. The streptavidin binds to the biocytin of the recorded cells, and the attached HRP, which stands for horseradish peroxidase, acts as a catalyst that breaks down hydrogen peroxide. Finally, the authors immersed the section in a solution of diaminobenzidine, DAB, including a bit of nickel, and a solution of hydrogen peroxide. This hydrogen peroxide reacts only in the recorded cells due to the HRP construct bound to the biocytin. This causes the DAB to precipitate within the recorded cells, filling up their entire dendritic and axonal branches. Importantly, the nickel in the solution also gets trapped in the DAB precipitate, making those cells visible in both light micrographs and ultrathin electron micrographs. The authors then subdivide the original 300 micron thick slice into several 80 micron thick slices. This is done simply to make the light microscopy tracing more accurate. Then these sections are stained and fixed again with osmium tetroxide and finally embedded in a plastic resin to prepare them for electron microscopy. The authors now trace the neurons in a light microscope using the software Neurolucida. Here is a clip from that same 2018 Journal of Visual Experiments paper showing part of that process. The researcher traces the neuronal processes in 3D by adjusting the depth of focus of the light microscope. Here is figure 1 of the Holler paper, showing a light micrograph stack of 2 recorded and DAB stained neurons. And here is the actual 3D tracing of those neurons using Neurolucida. The presynaptic axon is colored blue, and the postsynaptic dendrite is colored black. Using light microscopy, they found 3 possible synapses numbered on this figure. That is, they found 3 locations where the presynaptic cell's axon came close enough to the postsynaptic cell's dendrite that there might be a synapse, as far as the light microscope's crude resolution could tell. The authors then cut blocks around each of the putative synapses, 1, 2, and 3, and then ultra-thin sectioned each of these blocks at 60 nanometers thick, and collected the resulting series of these ultra-thin sections on TEM grids. These were then imaged in the transmission electron microscope and 3D reconstructed to determine if there was an actual synapse present. An actual synapse was found to be present in the first putative contact. So here is the light micrograph that the authors thought showed a synapse, but it was too low resolution to verify. They then sliced that block up and acquired electron micrographs like this. And in this electron micrograph, you can see that there is a presynaptic axon that is filled with a black substance, that's the DAB and nickel stain. There is a postsynaptic spine that is seen here, also filled with the DAB, and you can clearly see the synaptic junction, the synaptic cleft between the two. So looking at this electron micrograph, they were able to 100% verify that the close apposition that they saw in the light micrograph image was in fact an actual synapse. They also found an actual synapse was present for the third putative contact that they saw in the light micrograph. But the authors found no synapse for the second putative contact. Now, the authors were interested in more than just whether a synapse was present or not. They wanted to measure as precisely as possible the area of synaptic contact. They did this by carefully tracing the synaptic cleft and the synaptic densities in each ultrathin TEM section that cut through the synapse. Here is just one of those TEM sections. The authors took several TEM images of each section under different tilts so that they could align their view to see the synaptic clefts clearly. Then they spotted the subtle grayscale differences of the postsynaptic density. It's easier to see in an inverted image. And highlighted in red those areas that contained this PSD. The authors did this whole procedure of recording and light microscope reconstruction for many mice, but rejected all but 10 experiments, mostly due to incomplete filling of the neuronal processes with DAB. Here is figure 2 of the paper showing a different set of recorded neurons in a different mouse. And here is extended data figure 3. It actually shows each of the 10 experiments. Experiment 1, Experiment 2, Experiment 3, 4, 5, 6, 7, 8, 9, and 10. Each of these is a different mouse and a different set of recorded cells. This also shows all of the 16 in total synapses that were reconstructed during the experiments. Zooming in on one of these experiments, we see that in this experiment they recorded these EPSP responses. Then they found 2 synapses via electron microscopy. The first of these synapses was found at a distance of 89 microns from the cell body along one of the dendrites, and the other was found at a distance of 78 microns from the cell body, not necessarily along the same dendrite as implied by this simple picture. The EM reconstructed shape and area of the synapses PSDs are shown here, related to the 1 micron scale bar at the bottom of the figure. Now that we understand all the methods involved in the paper, let's get down to the real results. First off, they compared the 3D volume of each synapse versus its PSD area that they had painstakingly reconstructed. As has been well known from many previous studies, these two are well correlated. Spines with larger volumes have larger PSD areas. The presumed reason behind this often replicated correlation is explained in this excellent book by Sterling and Laughlin. I'll just read a quote from it here. In fact, to increase its synaptic strength, a synapse must enlarge. The presynaptic terminal enlarges to accommodate more vesicles and more active zones for release. The postsynaptic structure, typically a dendritic spine, also enlarges to accommodate more transmitter receptors, more synaptic scaffold proteins, and more regulatory proteins. In figure 3 here, the authors start to address the central question. Here the authors plot the mean EPSP amplitude, it's the white dots in this figure, recorded in each of the 10 experiments versus the summed PSD areas. To help understand this plot, here is the experiment associated with the uppermost dot in this plot. The sum of the PSD areas of the 2 synapses, shown here, is plotted on the x-axis, and the average EPSP amplitude seen in the recording is plotted on the y-axis. The gray dots above and below show individual EPSP trials. I want to make clear how crude this analysis is. It's ignoring where the synapses are on the dendritic tree, even though it is well known that synapses further from the soma will give correspondingly smaller EPSPs in this subthreshold regime that the experiment is being tested in. But even this crude measure shows a strong correlation of synapse size and physiological strength. But what we really want to know is how the synaptic current relates to a synapse's size. And for this, the distance of each synapse from the cell body must be taken into account. To do this, the authors used a computational model originally developed in this paper, which used dual patch clamps, one on the soma and one on the dendrite at a known distance. With this understanding of how EPSPs attenuate versus distance from the soma, they could interpret their EPSP amplitude that they measured at the soma and the distance of each synapse from the soma to infer the current injected by each synapse. And here is the result of that analysis. Each of the 16 synapses is shown in this plot. The x-axis shows the single synapse's PSD area, and the y-axis shows the estimated current injected by that synapse due to its AMPA receptor's opening in response to the presynaptic action potential. This is really the key result of the paper. This is showing that there is a linear relationship between the size of a synapse and its physiological strength. Finally, the authors analyzed their data to address the question of how many release sites there are per synapse. Recall that each of the 10 experiments recorded hundreds of EPSPs, and the amplitudes of the individual EPSPs could be plotted in a histogram like this. For example, the unusually large EPSP shown here would end up in a bin of the histogram over here. Let's develop a simple model of this under the assumptions of the multivesicular release model. Let N be the number of release sites, irrespective of how many physical synapses there are, and let P be the probability of each release site releasing a quanta of neurotransmitter following a presynaptic action potential. And let Q be the EPSP amplitude in millivolts due to one vesicle of neurotransmitter being released. In the drawing shown here, there are actually N equals 5 release sites. I wrote a little program with these variables and ran 10,000 simulated trials and got this plot. The 5 release sites are reflected in the 5 black bars in the histogram. Either 1, 2, 3, 4, or all 5 release sites can release a vesicle in any single trial. Now, this plot doesn't look very much like the plots in the Holler et al. Paper. But when I run the program with an amount of recording noise that they found during their experiments, they now do look very similar. The authors developed a model, a mathematical model, that could take the mean, standard deviation, and skewness of each histogram, and then estimate from it the N, P, and Q. They did this for each of the 10 experiments to estimate the number, N, of release sites in each experiment. For example, they estimated a whopping 16 release sites in this experiment that has only 2 physical synapses. They note that, crucially, our estimates of the number of release sites exceeded the number of synapses for all connections, on average by a factor of 4.4. The authors argue on the basis of this and other analyses that the data that they have collected effectively rules out the single release site per synapse hypothesis and favors the multivesicular release hypothesis. This multivesicular release hypothesis is actually the growing consensus of the field. And Holler et al. Authors relate their results back to single molecule localization studies, like this one here from Tang et al. 2016, that suggest presynaptic release sites and postsynaptic AMPA receptors are aligned into discrete transsynaptic nanocolumnar units that are inserted and removed as discrete units as the synapse strengthens or weakens. This is a very important topic that we'll have to wait to cover in another journal club. In conclusion, the Holler et al. 2021 paper developed a set of brilliant light and electron microscopy techniques that enabled them to relate the measured physiological strength of connection between 2 cortical neurons to the precise number, location, and sizes of their physical synaptic connections, as reconstructed by 3D electron microscopy. Through this, they found a linear relationship between synaptic size and physiological strength. This is a result that is crucially important to our ability to properly interpret the large-scale EM connectomics datasets that are now currently being generated. Thank you for listening.